Hello and welcome to my video for the Year 12 Research Methods revision for AQA A-Level Psychology. I don't quite know how many videos they're going to end up being of this because there's a lot to get through. So I'm going to try and get through as much as I can, as quick as I can, but still trying to be thorough, still trying to include all the AO1 and knowledge that you need. I'll drop in some of applications where I can, and there are the evaluations in there as well. I'm starting with this image that's on the screen, which is about something called dual coding, and this is something that's quite important, quite pertinent in teaching at the moment. Those that have already done the memory topic might be well aware of this. You might be aware of your visual spatial sketchpad and your auditory loop, which is included in the working memory model, which you need to know for the specification. What the theory of dual coding is when it comes to teaching is actually making use of these things and having an appreciation that actually when we encode something both visually, so in the form of pictures, and auditory, so in the form of words, that makes our memory trace stronger. So there are two things to anchor onto essentially, whereas if you've just got the auditory side of things in terms of sequencing, processing, the verbal information, then the way that you store that is linear. But actually if you've got the visual side of things as well, that really helps us to process that information as what's known as synchronously, which helps the information. So what I've tried to do in this video as well is try to include some icons and some images to try and help that memory trace and help you remember the research methods. We'll see how it goes, but that's why there are some icons as we go. So without further ado, let's get into the research methods. So before you get into lots of the research, there are certain terms that you need to be aware of, and we start generally with looking at the experimental method. And to understand the experimental method, you need to understand some of the terms that come before that, such as aims, which are the purpose of the study, hypotheses, which are a prediction, and these are ones where you certainly are going to have to be able to apply them. So the most likely way you get asked this is to write a directional, a non-directional, or a null hypothesis in an exam question. Key here is to make sure you're short and succinct and you include an operationalized IV and DV. So they are our next two terms. IV is the independent variable, the variable that's manipulated, and the DV is the variable you measure. And again, to operationalize is to define these things. So it's not good enough saying group A will have a better memory than group B. You have to operationalize what you mean by that. So group A might be the group listening to music, group B might be the group revising in silence, and when we say that their memory might be better, it might be the number of words recalled out of a number that are presented. So that's the difference between an IV and a DV, an operationalized IV and DV. We've then got extraneous and confounding variables, EVs and CVs. So an extraneous variable is anything other than the IV that could have an effect on the DV. These are something that you would wish to control before the experiment so that they don't have an effect. Confounding variables are variables that have had an effect on the DV, so they vary systematically with the IV, and it's something else that has changed the DV. It's then confounding your result. The word confound means to confuse, and obviously you wouldn't want those, so you'd want to control them before the study if you can to stop these confounders, or you'd have to do another study that tries to take these into account. Next, when we're talking about experiments, we look at something known as experimental design. And when we talk about design, this is how participants are put together in groups in the study. One of the most common mistakes I see made in research methods answers is people confusing research methods and research design. So a method is a way of gathering data, like an experiment or a correlation or an observation, whereas a design is how your groups are put together. There's only ever 3 designs that you need to know about in AQA research methods for psychology, and they are independent measures, repeated measures, and matched pairs. You do need to know what they are, you need to be able to apply them, and need to be able to evaluate them. So an independent measures design is where you've got separate groups of participants per condition of the study. And again, that's a common mistake. People say, oh, independent measures where you've got 2 groups. Well, actually, no, if you've got 5 levels of the IV, such as in Loftus and Palmer's car crash study, hit, bump, smash, collide, contact, then you need 5 different groups of participants. So that's why it's better to say a separate group per condition of the IV. And then you've got repeated measures, and this is where the same group of participants do all sides of the experiment, all levels of the IV. So you then repeat it. They do one, then the other. You could counterbalance these. We'll come to that later. So that's where half of the participants will do one half of the study first, then the other, and then the others would do the second one first, and then the first one. This gets over the order effects. We'll come to the evaluations in a moment. And then finally, you've got a matched pairs design, and this is where you've got two separate groups of participants, but in each group you've matched them up. You've almost tried to find a twin on a certain characteristics. That's characteristic. That could be age, it could be gender, it could be IQ, it could be any number of things. So if you've got a 30-year-old female with high IQ in one group, you'd want a similar person in the other group. So they are matched up. And that's trying to be the best of both worlds, where you've got different groups, but actually they're not too different that it's going to affect the results. So looking over to the strengths and weaknesses then, that could be the problems. Going back to independent measure design, it's great that you've got different participants. So what that means is it's more difficult to work out the aim of the experiment. There are no order effects, so they don't get bored or they don't get better naturally or get tired, so get worse, because they're only ever doing one condition of the study. So Loftus and Palmer again, they only heard one verb. If they'd heard all 5 verbs, they'd work out the aim of the study. However, there's problems here. You need more participants generally to do this sort of design, to get the same amount of participants in each condition or each level of the IV, and there are what we call individual differences here. So you might get personality characteristics that are affecting the results that you don't want there, whereas you won't get that in repeater measures because you're testing someone against themselves. And as you can see, looking at the strengths and weaknesses of the repeater measures, actually the evaluations, quite often this happens in research methods, they flip round. So a strength of a repeater measures is a weakness of independent and vice versa. So for a repeater measure, there are no individual differences. You're testing someone against themselves, so it doesn't matter whether they are good or bad at whatever your IV is or your DV is, so memory or aggression, whatever it may be, because you're testing them against themselves. You need fewer people because they're repeating the study and doing the other conditions, and you can counterbalance, as I said earlier. However, you're likely to get order effects. So if they did do the Loftus and Palmer study using repeater measures, then your results would have been the 5th time they'd seen that film of the car crash, it would have had an effect on them, and you would have got different results. So depending on your study and depending on what you're looking at, that might dictate which design you use. You can potentially get demand characteristics and you can potentially get fatigue as well. So what matched pairs is trying to do is have the best of both worlds. As I said, it's trying to get over order effects because you've got different groups, and also trying to reduce individual differences because you're trying to match people up. However, it takes more time and effort to find different groups of people and match them up on these criteria, and they're still not exactly the same, so there's still a chance of individual differences even though you've tried to control them. So it's all a bit of a balancing act. Next, we've got sampling. This is how participants are recruited into your study. 5 different types you need to be aware of. Random. This is where all members of the target population have an equal chance of being selected. Key here is that you obviously need to get all the names of people in your target population. So if it's in a certain school or college, then that's fairly straightforward. But if it's in a region or country, that's obviously a lot more difficult to get random sampling. So it's all down to your target population. Next is opportunity sampling. This is where you're using people that are there and available, probably one of the most common types of sampling in psychology. Professors using their psychology class, for example. Then you've got volunteer sampling. This is where you put up posters and ask people to come and take part in your study at a certain place or time, or you can email them out, and people who reply, those are the people that you use. You then got systematic sampling. This is where you choose, say, every 4th, 5th, 6th person, every nth person of your target population, but again you would obviously need a list of all the people in your target population. And then you've got stratified, and this is where you use a representative group, so you use percentage of key characteristics such as gender or religion or I don't know, job type. So if in your target population you've got, for example, 70% male and 30% female, in your sample of 10 you'd want 7 males and 3 females. So hopefully that's clear with what the definitions are. When it comes to strengths and weaknesses, the big thing you need to be aware of with sampling is you're looking for representativeness. And what that means is, do the people in your study represent the target population, the people you're looking for? And using these different methods generally has an effect on whether it's represented or not. So, for example, random sampling is seen as pretty representative because everyone potentially has a chance of getting picked, and so your sample should be quite representative of your target population. However, it's still not necessarily the case. You could end up by sheer fluke, because you're picking numbers out of a hat or using a computer program. You could end up with an unrepresentative sample based on your sampling method. Unlikely, but could still happen. It's time consuming. You may miss people. People might refuse to take part, and those could all affect how representative your sample is. Opportunity sampling, well, the strengths there are that it's easy, least expensive, least time consuming, and when you're looking at doing actual practical research, that's a really, really good thing. However, the weakness here is that you're probably going to end up with a biased sample. If they agree to take part in your study, then people who do that are going to have certain characteristics. They might have time on their hands. They might be willing to appease. Whereas you might miss people who don't like taking parts in studies and don't agree. But actually, that's quite important to again be representative. And so you've got a sample bias, could be unrepresentative. Next, you've got volunteer. The best thing about volunteer sampling is you can target people that you want to talk to. So, for example, if you're really doing a study on fat people, you can put your posters up in McDonald's or KFC or lots of places where fat people hang out, and they'll see your posters and they could take part in your study. So you can target your population, which is great. However, again, you're probably going to get volunteer bias or a biased sample, people who agree to take part or reply to posters or emails asking for people to take part have certain characteristics, and that could have an effect on your sample. For systematic, then again, you've got fairly representative. There's no chance of researcher bias here because you're picking every third person, which is great, or every fifth person. But again, is that going to be representative of your target population? Who knows? And then stratified sample, that's really good because, again, with the representation, it's more likely to be representative because you're picking certain characteristics to represent in your sample. Again, trying to avoid researcher bias. But again, you're going through all this hassle to try and get a stratified sample, try and represent it, but you're not going to be able to really truly represent how complexly different people are, and your sample will never truly represent the target population unless you test everyone in the target population, which again isn't going to happen. So again, it's all a balancing act. Nothing's perfect. Every study will choose one of these types, and they'll just be aware of the strengths and weaknesses of doing so. Next, we've got types of experiment. 4 main types of experiment you need to be aware of: lab, field, natural, quasi. The big thing that determines the difference between these, one, is where the IV is coming from, who's controlling, who's manipulating the IV, or is it naturally occurring, and then generally the environment in which it happens as well. So a lab experiment, the IV is manipulated by the experimenter and you've got good control over the environment. So a lab experiment is one that is highly controlled. A field experiment, the IV is still manipulated by the experimenter. What I mean by this, if you were testing the difference between drinking tea, coffee, water, and Red Bull on word speed production, the experimenter would be the one telling individual people, right, this is what you're drinking. So in a lab experiment, that would happen in a really controlled environment. In a field experiment, the experimenter could still tell the different groups of participants what they're going to be drinking, but it would happen in a more natural environment, such as a work environment or in a school. A natural experiment, to do that same study as a natural experiment, the IV is naturally occurring. So what I mean by this is you would say, right, who drank tea this morning? And they would be the tea group. Who drank coffee? They would be the coffee group. Who's had Red Bull? And they would be the Red Bull group. So the IV has already happened. The experimenters had nothing to do with who's drunk what. And then finally, we've got a quasi-experiment. And this is where the IV is fixed. These tend to be cases where there's nothing you can do about, I don't know, the gender you were born with. Let's not get into that. Or your eye colour. Those things are fixed. And so if you wanted to test those sorts of conditions, the key thing here with the quasi experiment, you could still have an IV that is gender but test that in a controlled environment. So a quasi experiment could happen in a controlled environment. The environment's not as important here. It's all to do with the IV being fixed and naturally occurring, not just like a natural experiment where something's happened to for it to occur. For a quasi, it's already happening. So strengths and weaknesses here. Generally, it goes down to control. And that then has an impact on internal or external validity. So for a lab experiment, there's high levels of control. So a strength of a lab experiment is that you've got high internal validity. You should be fairly confident that the effect on the DV is only being caused by the IV in a lab experiment. Not always the case, but you'd be more confident because you've got control over the IV, you've got control over the environment as well. However, weakness of a lab is that is then going to lead to a lack of external validity. It's quite an artificial way to look at things. So say again in my drink experiment, I've got them to come into the lab 3 days before I'm doing which drink has an effect on how many words they say, and they've had the same amount of sleep, they've had the same diet for 3 days, so I've controlled everything. That's great. But actually in the real world that doesn't happen. People drink different things, people eat different things, people sleep a different amount, and so it's not what we call externally valid. It might not actually happen that way in the real world, and that's really important for psychology. Whereas in a field experiment, you're probably going to have better external validity because you're testing that in a classroom or in an office where people have had different meals and different amounts of sleep. Then it might be a bit better. So you've got better external validity, improved external validity, but potentially you're losing then some control. You are losing some control, so you potentially got poorer internal validity. Oh, I've got that wrong there. That should be poorer internal validity. Natural experiment is so generally better still, high external validity because the IV is naturally occurring, the environment tends to be natural as well, allows you to study topics that are hard to control. So an example like that might be the effect of adoption. So, as an experimenter, you're not going to get a group of kids and say, right, you are going to be adopted and you're not. Highly ethical, questionable there. That wouldn't happen. But you can test kids that have already been part of adoption. So it allows you to test things that are hard to test anyway. However, the problem with that is that there's potentially then more confounding variables. We don't know what has led to that. Have they had a poorer home upbringing in younger years? So whatever effect we're trying to test, attachment for example, it's hard to tease apart. Well, what's the impact of the adoption rather than other characteristics. Also hard to replicate as well, so you lose out on reliability. And then finally, quasi can take place in controlled conditions, which is great, but again you haven't got that random allocation, and so other things can affect whatever it is you're testing. So if you're testing, I don't know, the gender difference on male or female on reaction time, well, there are a number of things. Obviously society treats boys and girls differently, and so actually how much of it is to do with whatever your hypothesis is and how much is to do with socialization or the biological impacts, and so it's hard to control. Moving on, let's talk about data, because that's what you get at the end of an experiment. Lots of different terms you need to be aware of if you haven't figured that out already. What's wrong with you? There's lots of terms that you need to be aware of in research methods. So first kind of classification of data is primary or secondary. So primary, you probably already know this from primary school anyway, is first-hand data that you collect yourself, but secondary is pre-existing. The impact of this in psychology, what you need to be aware of is that the best thing about primary data is that it is then specific to the aim of the study. So you are studying what you intend to. However, it can be harder to get primary data, so that's a weakness. Whereas secondary data is pre-existing, so it's already out there, which is great. So I could do a study on using secondary data now if I wanted to. However, it's not specific to the aim. Not with an E. Why have I put an E? An aim. Next is qualitative and quantitative data. So qualitative data is non-numerical data. Generally, it's written data, but it can be pictures, drawn pictures, photographs, or quantitative data, so that's numerical data, numbers. So the benefit of qualitative data is you tend to get rich, more detailed information. So if it's an interview, for example, or a diary entry from someone about the topic you're studying, that's going to be rich information, which is great. However, it's probably going to be harder to compare, harder to replicate, and harder to show your results. Whereas with the quantitative data, it's going to be a lot easier to compare. You can show charts, you can show graphs, and represent that data really, really easily. However, you're then missing out on that rich, detailed information. It lacks detail, it lacks nuance. You can't really tell the whole story. What you can then do though is you can represent that data. So mean, median, and mode together are known as measures of central tendency. And they are all types of average, and they can represent your quantitative data. So the mean is you add all the numbers together, divide by the number that you've got. The median is the middle number, middle finger, and the mode is the most common number that you get. But again, you're probably aware of all those terms from year 7, but actually you need to be aware of the strengths and weaknesses of using them. Maybe you're not as up with that, and that's quite often required in psychology. So the best thing about the mean is all scores are taken into account, so it's really representing the data. However, your mean is affected by extreme scores, so if you've got an outlier, your mean won't be accurate. You wouldn't be able to use it. You'd have to use a median or a mode instead. And that's the strength of both of those. So median and mode are less affected by outliers, but the weakness there for them is that they then do not take all the scores into account. And so are you fully representing the data? After you've got your measures of central tendency, you've then got measures of dispersion, which are your range and your standard deviation. So this shows how spread out the scores are. The average alone doesn't give you everything you need. You then need to know, well, what does the data actually look like? Did everyone get that? Well, no. How many and how far apart was it? And that's what your range and standard deviation can tell you. So the range is your largest number minus your smallest number. Really easy to calculate, but again can be influenced by extreme scores. Whereas your standard deviation, you'll never have to calculate this in a psychology exam, but you do need to know what it is. You do need to be able to read a table that's got a standard deviation in. In simple terms, it's the average distance from the mean. So a larger standard deviation means the scores are more spread out. So for a psychological experiment, what it generally means is that the IV has had a different effect on different people in different ways. So for example, say it was watching a horror film versus a comedy and the number of nightmares that causes. If you've got a large standard deviation there, what that means is that some people are having more nightmares and others aren't. More importantly, though, in psychology, if it's a treatment that you're looking at, say you've got 2 treatments, CBT and drug for OCD, and they both have a similar mean, similar average improvement score in OCD, but one, say the CBT, has a larger standard deviation. What that means is it works for some people and doesn't work for others. So that's quite significant, and that's something to know. And that's why we need both a measure of central tendency, an average, and a measure of dispersion. So the best thing about standard deviation, it does take all scores into account. It is harder to calculate, but don't worry, you wouldn't have to do that. And then let's just tack on peer review here. You'll need to know more about that in year 13, but for year 12, all you need to really be aware of is that peer review is a way of having your research checked by other people. Once a study's done, it will be written up and it will be sent to journals and journal articles for reviewing by different people. Generally, these people are experts in their area. They are your peers and they are reviewing it, hence peer review. So what they do is they check for the quality of the research. They suggest any amendments that might need to be made. They can either accept it as it is, they can accept with corrections, or they can reject it, and it can allow allocation of funding. So if you're known as a good researcher and you're getting lots of published research, you're then probably more likely to get research funding in the future. It's not perfect, however, because the reviewers tend to be anonymous, so they could push their own agenda. Say that you've come up with a new theory and your reviewer's got a theory that goes against what you're saying and it's their theory. Well, they might not like that. You'd like to hope that a scientist would have the integrity to admit that, but not always. There's also publication bias. This means that research that is found to be significant is much more likely to get published. That means that we've got what's known as the file drawer effect. So lots of research has been done, not found to be significant. Well, that is as important research. It's good to know when things haven't worked or what's not there, because we can refine hypotheses after that, and actually it would stop people then maybe repeating studies that have already been done. But yeah, we only ever see the research that is significant generally. That means when you're doing a meta-analysis, which is where you look at lots of different studies, you're only going to generally find studies that have found a result, whereas there could be more hiding behind that haven't found a result, and so we're not generally getting an accurate representation. And breathe. That's about it for part one. It would be worth you maybe doing some exam questions on experiments and sampling and designs. Just test yourself, see how much you've done on there, go over anything that you need to. I will do another video looking at the other methods, so observation, self-report, etc., etc. Hope you found it useful. Thank you.